Hey everyone, and welcome to This Explainer. If you're an MSc Zoology student staring down the barrel of that high-stakes Semester 4 Vivivos, you are in the exact right place. You've spent years studying these incredible creatures, but let's be real, acing your final exams takes way more than just memorizing a textbook. You've got to understand the structural logic, dodge those tricky evolutionary traps, and really grasp the biological why behind insect anatomy. So today, I'm going to act as your professor, distilling the absolute highest yield concepts from your advanced systematic syllabus into a masterclass that's going to make your final review super efficient and totally unforgettable. Let's dive in. Okay, let's quickly run through our roadmap for this session. First up, evolutionary origins and basics. Then, complex anatomical distinctions. Third, the why of mechanisms. And finally, applied systematics and biodiversity.
Right, let's kick things off with Section 1, Evolutionary Origins and Basics, or as I like to call it, the Traps of Apterygotar.
We're starting right at the foundation of your syllabus, the Apterygoda. Since these primitive hexapods never evolved wings, they hold on to some really tricky ancestral secrets. Picture this. You sit down for your viva, and the examiner hits you with this classic trap. Which primitive order is characterized by an absence of both antenna and eyes? Think about it. Almost every insect you can picture has them, right? It really forces you to look at the very base of the evolutionary tree.
Well, the answer is Protora, and the biological logic here is fascinating. Since they lack true antennae, they actually compensate by using their front legs to sense their environment. I mean, they are literally walking around on their middle and hind legs, holding that front pair up like makeshift TV antennas. It's an absolutely perfect example of an organism adapting when it doesn't have standard sensory organs.
Now, speaking of evolutionary oddities, take a look at the Ephemeroptera, or mayflies. They have this unique subimago stage. This is literally the only instance in the entire class Insecta where an insect molts after its functional wings have developed. Literally every other insect order out there, once the wings pop out, the molting stops permanently. So definitely keep this transition phase in your back pocket when your professors ask you about insect development and metamorphosis. Moving right along to Section 2, complex anatomical distinctions. This is all about surviving those overlapping characteristics that trip everybody up in the viva. So, when you hit Unit 3, you're going to be bombarded with microscopic differences that completely change an insect's classification. Take lice, for example. If your professor asks you to distinguish between the two major lice orders, Anaplora and Malaphaga, please don't just guess based on where they're found. You absolutely have to focus on the anatomy of the head, and specifically how that relates to what they eat. The comparison here boils down to brilliant biological logic. Look at the Malaphaga, the chewing lice. They've got these broad, wide heads. Why? Because they literally need the physical real estate to anchor the massive biting muscles required to chew through tough skin and feathers. But then you look at Anaplora, the sucking lice. They just have these narrow little heads because all they're packing inside is a tiny, highly efficient blood-pumping mechanism. It's form-following function to a T. Let's look at another classic split that tests your anatomical vocabulary. Telling a caterpillar from a grub isn't just about saying, one's going to be a moth, one's going to be a beetle. You need the technical terms. A caterpillar from the order Lepidoptera has these fleshy abdominal prolegs equipped with microscopic hooks called crochets. A grub from the order Coleoptera, it completely lacks those abdominal prolegs, and instead it's defined by a heavily sclerotized, really well-developed head capsule. And to quickly summarize the high-yield diagnostic features of the Orthoptera, your grasshoppers and crickets, just remember their diagnostic triad. Their leathery forewings, technically called tegmina, aren't for delicate flying. They're tough armor protecting the membranous hindwings and the soft abdomen when they jump into rough bushes. Combine those tegmina with the tymptatum for sound production and their saltatorial or jumping hind legs, and boom, you've completely nailed their diagnostic profile. Okay, let's jump right into Section 3, the why of mechanisms, because let's face it, rote memorization is not going to save you in a high-level exam. You really have to understand how these biological structures actually function in the wild to guarantee survival. Take the columbola, commonly known as springtails. Their whole survival strategy is based on the furca. It's a simple, brilliant three-step escape hatch. First, it's held under intense physical tension by a catch mechanism called the tenaculum. When danger gets close, the tenaculum releases, the furca snaps forcefully against the ground, and it instantly catapults the tiny insect away. It's basically built-in, lightning-fast kinetic energy. Now here is a trap I see students fall into all the time with aphids. You look at an aphid and see those two little tubes on its back end, the cornicles. It is super tempting to say, oh, that's where the honeydew comes from. Actually, scratch that. Honeydew comes from the digestive tract. Those cornicles are twin exhaust pipes dedicated entirely to chemical defense. They pump out alarm pheromones and sticky, waxy fluids to blind and distract predators like ladybird beetles. Don't mix those up. Next up, let's talk about the true flies, order Diptera. Have you ever wondered how they manage to balance perfectly in midair? You see a housefly pulling off hovering maneuvers that would make a fighter pilot jealous. But the biomechanics required to achieve that kind of stability are insanely complex and rely on a really wild evolutionary modification. And the secret lies in the halteres. In Diptera, the hind wings didn't just disappear. They evolved into these tiny vibrating knobs. As the fly zips around, these halteres experience what physicists call the Coriolis force. Sensors at the base detect those minute physical forces in real time. They are literally built-in organic gyroscopes, telling the fly's brain exactly how its body is rotating across 3 dimensions. It's why Diptera are basically unmatched in aerial agility. Now compare that to Hymenoptera, the bees and wasps. They also need incredible flight efficiency, but they solve the problem completely differently. They use homuli. These are microscopic hooks that essentially zip the forewing and hindwing together while they're flying. By locking 2 smaller wings into one massive, powerful wing surface, they drastically cut down on air turbulence, making those long foraging flights way more aerodynamically efficient. Finally, let's bring it all together in Section 4, Applied Systematics and Biodiversity. Systematics is definitely not just about sorting dead bugs in a dusty museum drawer. It's the absolute foundation for critical fields like medicine and forensic science. Take the beetle family Dermastidia. Forensic investigators rely entirely on the feeding habits of these exact beetles to date crime scenes. Because their diet is basically just dry skin and tough keratin, they're known as late-stage colonizers. They won't touch a fresh scene. So if an investigator finds them, that biological clock instantly tells them the death happened months, not days prior. Or look at medical entomology with the order Siphonaptera, the fleas. Their anatomy is just terrifyingly perfect. Notice that extreme lateral, side-to-side compression? That flat shape is an evolutionary masterclass for parasites, letting them slice right through dense rat fur without getting snagged. Add in a pleural arch packed with elastic resilin for massive jumps, and you have a hyper-efficient vector perfectly designed to transfer deadly pathogens like the plague from rodents to humans. Now, shifting gears to biodiversity, if you get asked about India's unique entomological richness, there's one specific number you need to remember: 3. India is a megadiversity hotspot because it sits right at the junction of 3 distinct biogeographic zones: the Afrotropical, the Indo-Malayan, and the Palearctic regions. And it's exactly this massive geographic overlap that creates such wild evolutionary phenomena. You've got Himalayan, tropical, and desert species all interacting on a single subcontinent. For example, look at the common mine butterfly in India. It has evolved to perfectly mimic the appearance of the highly unpalatable common tiger butterfly. It's an incredible visual disguise entirely driven by the diverse predator pressures of that overlapping habitat. Lastly, let's touch on economic entomology. Your professor will almost certainly ask you about our endemic resources, so we really need to zero in on the physiology of the moth family Saturnia, specifically in the state of Assam, to understand the treasure that is Muga silk. So, Muga silk is produced exclusively by the species Anthorea assamensis. Because of this endemic moth's highly specific physiology and local diet, it spins a silk that is naturally a shimmering golden color, and it is completely unbleachable. This just proves that mastering systematics isn't just a classroom exercise. It has direct, real-world ties to massive global agricultural economies. All right, as we wrap up this explainer, I want to leave you with a bit of a provocative thought. We rely heavily on physical anatomy for systematic classification, sure, but we also use behaviors, like the incredibly complex honeybee dance, to classify social insect species. So, if our classification relies on these delicate environmental and behavioral cues, how might the rapid climate change that's actively rewriting insect behavior force us to completely redraw the entire evolutionary tree in our lifetimes? I'm going to leave you with that thought to chew on. Keep questioning the grand design of nature, trust your anatomical foundations, and best of luck on your Semester 4 Viva Voice.